I would like to reintroduce Rebecca Shipley, who's an early career scientist at APHA, who's going to talk about the case investigations across the UK in companion zoo and wildlife animals. Thanks, Rebecca.
Thanks, Sharon. I'm just gonna... Can everybody see that okay?
Yep, that's fine. Thanks very much.
So yeah, I'm Rebecca Shipley. I recently completed my PhD at the APHA and the University of Sussex, and now I'm working as a coronavirus virologist, still in the virology department. I'm looking at sort of surveillance, and we're doing a few in vivo studies to look at transmission of different virus variants as well. But today I will be talking about SARS-CoV-2 cases in UK companion zoo and wildlife animals. So I'll start off talking about our surveillance in wildlife. Sharon briefly mentioned that we work in a collaboration under a COVRIN research project, and this is where we take opportunistic samples from wildlife to look at SARS-CoV-2 prevalence. So I'll just do a brief introduction. Why are we interested in SARS-CoV-2 in wildlife? Obviously, it appears that SARS-CoV-2 originated in wildlife, and there's a potential for emergence of wildlife reservoirs and spill back to humans. There's also potential for evolution of new variants with different characteristics, and this would obviously have implications for effective control of disease in human populations, but it would also have serious impacts on wildlife management, research, rehabilitation, and conservation activities. So given the widespread infection of humans, we obviously need to plan for the implications of infection in wildlife as well. So Fabian did a good overview of what species might be susceptible, and obviously he's done quite a big investigation of the host cell receptor and the presence of that in a range of potential hosts. So bats, rodents, carnivores, cetaceans and primates. But obviously, as Fab's highlighted, presence isn't always confirmatory, and the distribution of receptors may influence susceptibility. But obviously we do have the experimental infection studies which have identified a range of susceptible hosts such as cats, dogs, ferrets, mink, hamsters, raccoon dogs, white-tailed deer, bank voles and deer mice. And obviously these studies have shown various levels of virus replication and shedding, and some have shown transmission as well. But obviously experimental studies don't always mimic natural conditions. We have also seen transmission to free living wildlife, so the mink farms in the US and other areas of Europe, the white-tailed deer in the US, as Fabian mentioned, badgers in France and pine martens, and there was also a leopard in India as well. So the way we collect our sample is predominantly opportunistic. So this is sampling off the back of other research programs or wildlife management activities, and we have highlighted priority species groups, so mustelids, canids, cervids and rodents, and the samples that we typically take are throat and nasal swabs to detect the presence of viral RNA. We also try and take blood samples to detect the presence of SARS-CoV-2 specific antibodies. And we've also received droppings from bat roosts, again, to detect the presence of viral RNA. And we've also actually received various tissue samples as well. So this is just a few examples of the samples that we've received. We've received a number of fox and badger samples, and also American mink samples, which are an invasive species. So we had help from various wildlife trusts to receive those samples, and also stoats as well. We also have contacts, I believe, in the New Forest with wild boars, but those samples are yet to be collected. And also with the help of the National Trust, we've managed to take a number of samples from different species of deer as well. And then finally, we have received a number of bat droppings from lesser and great horseshoe bat roosts. And we also received bats as part of our rabies surveillance activities. So we've been taking throat and rectal swabs from some of those as well. And we've also received a number of grey squirrel samples as well. So this is just an overview of everything that we've sampled and tested so far. So as you can see, we've tested against our SARS-CoV-2 e-gene PCR, none of which have come up positive so far. And we've also managed to test a number of badger serum samples on SARS-CoV-2 ELISA and SARS-CoV-2 VNT, again, which none have come up positive. We also plan to screen all of these samples against a pan-CoV PCR as well, just to see if there's any other coronaviruses circulating in these species. This is still very preliminary, and we've only tested some bats, of which have all been negative so far. So this brings back to sort of our dynamic framework for surveillance and risk assessment, and this just has been extracted from Richard Delahay's publication, and it gives an overview of identifying priority species to target. So we're at the stage of taking opportunistic sampling. So we've already sort of assessed the likelihood of exposure in a number of wildlife species, and then we've looked at host susceptibility by looking at previous experimental infections or naturally acquired immunity infection, and then looked at the likelihood of maintenance and onward transmission. And this is essentially a feedback loop. So if we do identify a potential reservoir, then we'll probably hone in on that a bit more as well. So I'll now just move on to surveillance in companion and zoo animals. So SARS-CoV-2 in animals became a reportable disease in the UK in February 2021. And so as a result, APHA was designated the National Reference Laboratory for SARS-CoV-2 in animals. SARS-CoV-2 infection has been detected in dogs, cats, ferrets and hamsters, and a number of zoo animals, so otters, large cat species, gorillas, binturong and hyenas. So we do offer primary testing for SARS-CoV-2 for non-human primate material. This isn't typically for antemortem testing of companion animals such as dogs, cats and mustelids. I'll give an overview of the process for investigation on the next slide. But yeah, in general, the testing for the non-human primates would be undertaken in a class 3 MSC in high containment. So we've mainly done confirmatory testing for SARS-CoV-2 at the APHA, and that covers dogs, cats and mustelids, and primary diagnostic testing is usually carried out by the PVS or a private lab, and then any positive results would then be reported to the APHA, where any cases are triaged using a questionnaire, and then, if applicable, referred on to the APHA One Health team. We usually organise follow-up sampling, which can in some cases be time limited, and then we organise an RT-PCR on the original swab if it's still available, or follow-up samples. And obviously any negative results will just be reported back to the PVS, and positive results are reported to the One Health team. So I'll now just give an overview of some of the cases that we've had at the APHA. Our first one was in June 2020 in a pet cat. This cat was from a COVID-19 positive household and initially demonstrated respiratory signs. The PVS collected oral swabs, and SARS-CoV-2 viral RNA was detected at a private lab. So the One Health team organised follow-up investigation, and additional samples such as bloods and more swabs were taken, and those and the original samples were transferred to the APHA. So we confirmed presence of SARS-CoV-2 in the original swab sample, and whole genome sequencing. A whole genome sequence was generated from the sample, which confirmed an early SARS-CoV-2 lineage. And then we followed up with a virus neutralization assay on the blood sample, and it did indeed detect specific SARS-CoV-2 antibodies. Interestingly, there was another cat in the household, and that actually tested negative for SARS-CoV-2, and the cat and the owner made full recovery in this case. So our next case wasn't until a year later in June 2021, where a poodle Shih Tzu cross from Jersey presented to the PVS in a collapsed state with severe respiratory infection. The dog was euthanized the same day, and the primary veterinary surgeon took combined conjunctival and oropharyngeal swabs. It was again from a COVID-19 positive household, and the SARS-CoV-2, there was SARS-CoV-2 RNA detected by a private lab in Germany. So the One Health team organised the residual sample and the cDNA to be transferred to APHA, where again it was confirmed. And in this case, the whole genome sequence was obtained and confirmed to be the Delta variant. Later that year, in November 2021, we had another Labrador retriever, in this case a dog, and it presented to the PVS with sudden onset of rapid breathing and raised temperature. So the PVS took dry conjunctival and fecal swabs and one oropharyngeal swab in VTM, and actually tested this dog using a lateral flow test where it was SARS-CoV-2 antigen was detected. Again, it was from a COVID-19 positive household. So APHA, the One Health team, organised the testing of the swabs, and it was confirmed positive in the oropharyngeal swab. And in this case, we actually isolated virus from the oropharyngeal swab sample. And again, the whole genome sequence was confirmed to be the Delta variant. So just a month later, we also had another Labrador retriever dog from a COVID-19 positive household with gastrointestinal signs, myocarditis, and it unfortunately passed away during a scan. SARS-CoV-2 viral RNA was detected by a private lab in a pharyngeal swab. And then additional swabs plus the original pharyngeal swab and the carcass was provided to APHA, where we confirmed the presence of SARS-CoV-2 viral RNA, and we also isolated the virus from the bronchoalveolar fluid as well. Again, this was confirmed to be the Delta SARS-CoV-2 variant. And then this brings me along to my last case. So initially, there was a male Amma tiger which presented with acute onset of coughing and lethargy. And so the PVS obtained a sputum swab, and it was confirmed to be positive by a private lab. So additional samples were collected from the male tiger and 2 further female tigers, which also showed similar clinical signs. And SARS-CoV-2 viral RNA was detected in all samples from each of the animals. So all 3 animals in this case did have SARS-CoV-2 at some stage. We also received a sample from a male snow leopard, which was from a different cage, but it had shown similar clinical signs. However, no SARS-CoV-2 viral RNA was detected. And again, the whole genome sequencing was obtained from all 3 tigers and confirmed to be the SARS-CoV-2 Delta variant. So this is just a case summary of all the cases that I've just explained. And as you can see, the majority of the cases were a Delta variant, with the exception of the one early case in 2020. So as well as our NRL responsibilities, we've also taken the opportunity to test sera that we receive as part from the rabies pet passport scheme. Currently, we've only tested cat sera, and we've tested about 176 samples, where 11 of which were positive on our SARS-CoV-2 ELISA. So any positives we hope to confirm by VNT as well. And that brings me on to the next steps. So we'll obviously continue our wildlife surveillance. So we'll continue to test on our SARS-CoV-2 e-gene PCR and follow up with our pan-coronavirus PCR as well. And this would hopefully highlight more high-risk species. And we also want to continue our sera surveillance of pet sera. So what we're in the process of doing is actually taking the sera from backdated from the beginning of the pandemic and testing anything that we've received from the UK on an ELISA that we've until present day. And that will hopefully give us a good understanding of sort of the prevalence of cats and dog seroprevalence in the UK. And then hopefully test any positives on VNT using the different SARS-CoV-2 variants. And obviously we will continue our NRL responsibilities of processing any suspect animal SARS-CoV-2 cases, and then with the aim of developing a risk assessment approach going forwards. I just wanted to say thank yous to the virology department at the APHA, as well as the One Health team, which orchestrates all of our diagnostic submissions, and also Richard Delahaye and Ruth Cox at the National Wildlife Management Centre, who orchestrate obtaining the wildlife samples as well. And that's it from me. Thank you very much.